Hello friends, how are you? Hope all are fine. Welcome to Sam Biology class. Today we shall discuss about chromosomal mutation. Before that, may I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Chromosomal mutation, also known as chromosomal aberration, refers to the large-scale variation involving a change in chromosome structure, change in the number of whole chromosomes or whole chromosome sets. Chromosomal mutation can be grouped into three basic categories. They are: number one, chromosome rearrangements. Number two, aneuploidy. And number three, polyploidy. First one is chromosome rearrangements. It is a type of mutation which alters the structure of chromosomes. For example, a piece of a chromosome might be deleted, duplicated or inverted. Second type of chromosome mutation is aneuploidy, which alters the number of chromosomes. Here, one or more chromosomes are added or deleted. And the third type of chromosomal mutation is polyploidy, where one or more complete sets of chromosomes are added. Many organisms which possess a single set of chromosomes most of their life cycle are known as haploids. For example, yeast. Human beings and other organisms which possess two sets of chromosomes are known as diploids, and several other organisms which have more than two sets of chromosomes are known as polyploids. First one is chromosome rearrangements that change the structure of individual chromosome. There are four basic types of arrangement, such as A. Duplication, B. Deletion, C. Inversion, and D. Translocation. First one is duplication, in which a part of the chromosome has been doubled or duplicated. That means a particular segment is present as two copies. Consider a chromosome having the segments A, B, C, D, E, F, G. When duplication includes the segment DE, then after duplication, the resulting chromosome will have two copies of DE segments. So such type of rearrangement takes place after duplication. Here you can see that two such segments EF are present. This is one copy and this is another copy. There are different types of duplication. When the duplicated region is located immediately adjacent to the original segment, then such type of duplication is known as tandem duplication. This is an example of tandem duplication because the duplicated region is immediately adjacent to the original sequence. In some other cases, the duplicated region is located at some distance on the same chromosome or another chromosome, and that type of duplication is known as displaced duplication. Displaced duplication. So in this example, we will see how the chromosome rearrangement takes place due to displaced duplication. These are the centromeres, and here it is A and B. So due to displaced duplication, the chromosomes will be like this. Here it is CD, and next one is EF. This is the original sequence, and after duplication, here it will be G, and the duplicated region will be located at some distance from the original segment. So such type of duplication is known as displaced duplication. After duplication, the duplicated region may have the same orientation as that of the original sequence, or it can be inverted. If the duplicated region is inverted, then that type of duplication is known as reverse duplication. Duplications may have major effects on the phenotype. For example, in the fruit fly Drosophila melanogaster bar eyes, in which there is a reduced number of facets, making the eye small and bar-shaped, arise due to a small duplication on X chromosome. When an individual is homozygous for a duplication, then because both the homologous chromosomes of a pair carry the duplication, no problem arises during pairing in prophase 1 of meiosis. But in case of heterozygote, because one chromosome is normal and the other homologous chromosome of the pair carries duplication, loop formation takes place during prophase 1 in meiosis in order to ensure pairing between two chromosomes. So, in case of heterozygote, one chromosome is normal and the other chromosome carries the duplication. So, during pairing, the duplicated regions form a compensation loop so as to ensure pairing between the two homologous chromosomes. So, this figure shows formation of compensation loop during pairing in prophase 1. Second type of chromosome rearrangement is deletion, in which a part of the chromosome is lost or missing. Consider a chromosome having the segment AB and C, D, E, F and G. When it undergoes deletion of the segment DE, the resulting chromosome will be like this. The resulting chromosome will be short as compared to the original chromosome. So here it is C, F and G. This segment has been lost because of deletion. Deletion is of two types. One is terminal and the other one is intercalary. In this example, for the segment DE to be lost, this chromosome must break in two places. So when these chromosomes break in two places, the resulting segment is lost and the remaining segments are joined. Such type of deletion in which a middle segment of the chromosome is lost is known as intercalary deletion. In some other cases, the terminal segment can also be lost. Such type of deletion is known as terminal deletion. So here it is A, B, C, D and E. This terminal segment has been lost, so this is the resulting chromosome. A large deletion can be detected easily because the resulting chromosome is noticeably shortened. In an individual homozygous for a particular deletion, because both chromosomes of the homologous pair carries the deletion, no special problem arises during meiosis. But when the individual is heterozygous for a particular deletion, because one chromosome is normal and the other chromosome is having the deletion, so problem arises during pairing in prophase 1 of meiosis. For pairing to take place, the normal chromosomes form a loop known as deletion loop in order to ensure pairing the two homologous chromosomes. Because one chromosome is normal and the other chromosome has the deletion, so during pairing, the normal chromosome form a deletion loop in order to ensure pairing between the two homologous chromosomes of the pair. So here it is like this. In some cases, a particular segment of the chromosome is inverted, that means it turned by 180 degrees. Such type of chromosome rearrangement is known as inversion, which is of two different types. Number 1, paracentric inversion. Number 1, paracentric inversion, and number 2, pericentric inversion. Pericentric inversion. The inversion in which the centromere is not included is known as paracentric inversion, and if the centromere is included, then it is known as pericentric inversion. Consider a chromosome having segments A, B, C, D, E, F and G. For example, if the segment DE is inverted, then the resulting chromosome will be like this. The sequence will be AB, then C, then the segment is inverted, so it will be ED, following which it will be F and G. So in this process of inversion, centromere is not included. Such type of process is known as paracentric inversion. But when the centromere is included, for example, the segment BC is inverted, then the resulting chromosome will be like this. So in this case, the sequence will be AC and then B, then it is normal DE and FG. So in this second example, the segment BC is inverted. So in this process, the centromere is included. Such type of inversion is known as pericentric inversion. Pericentric inversion. Inversion heterozygotes are found in many organisms, for example, in a number of plants, some species of Drosophila and mosquitoes and grasshoppers. Inversion may have played an important role in human evolution. If an individual is homozygous for a particular inversion, no special problem arises during meiosis because in both chromosomes of the homologous pair, the segment has been inverted, so no problem arises during pairing. But when the individual is heterozygous for the inversion, problem arises during pairing. This is because one chromosome is normal and in the other chromosome the segment has been inverted, so pairing do not take place normally. In order to facilitate pairing between the homologous chromosome, inversion loop is formed. This figure shows the formation of inversion loop. In this homologous pair, this is the normal chromosome and the other chromosome has the inversion in which this particular segment has been inverted. Because of this inversion, normal pairing is not possible. In order to ensure pairing between the homologous chromosome, inversion loop is formed. As a result, pairing can occur between the non-sister chromatids of homologous chromosome that results in crossing over. Finally, there is another type of chromosome rearrangements known as translocation, in which genetic material is moved between non-homologous chromosomes or within the same chromosome. Translocation should not be confused with crossing over, which is the reciprocal exchange of genetic material between homologous chromosomes. Translocation can be classified into two different groups. One is non-reciprocal translocation, and the other one is reciprocal translocation. In non-reciprocal translocation, genetic material is moved from one chromosome to another non-homologous chromosome. This is the one-way transfer of genetic material from one chromosome to another non-homologous chromosome. For example, A and B are two non-homologous chromosomes. So this is the chromosome A. So this is the chromosome A, and the sequence is A, B, C, D, E, F and G. And the other chromosome is like this: M, N, O, P, Q, R and S. If the segment DE is transferred from chromosome A to chromosome B, then this type of translocation will be called as non-reciprocal translocation, and in this case the resulting chromosomes will be like this. So here these are the centromeres and the sequence is AB, next one is C, F and G, and the other chromosome is like this. Here it is M, N and other R is O, P, next D and E, then Q, then R and then S. So in this process, the segment DE is transferred unidirectionally from chromosome A to chromosome B. In case of reciprocal translocation, the transfer of genetic material is reciprocal. For example, the segment DE is transferred from chromosome A to B, and the segment PQ is transferred from chromosome B to chromosome A. So there is a two-way transfer of genetic material between chromosome A and B. Because of this reciprocal transfer, the resulting chromosome will be like this. So here it is AB, next one is C, and then P, then Q, and then F and G. And the other chromosome is MN, next O, after that DE, next R and S. So these are the two resulting chromosomes formed due to reciprocal translocation. Deletion frequently accompanies translocation. Sometimes whole arms fusion takes place between non-homologous chromosomes through translocation. For example, these are two acrocentric chromosomes. They are non-homologous. If these two chromosomes have undergone breakage at two different locations, then because of this breakage, the two long arms can be joined to a common centromere that will generate a metacentric chromosome with two long arms and another chromosome with two very short arms. The smaller chromosome often failed to segregate during cell division, and this caused a reduction in the number of chromosomes. Such type of translocation is known as Robertsonian translocation. And Robertsonian translocations are the cause of some cases of Down syndromes. This process might have played an important role in the evolution of human beings. The effect of translocation on chromosomal segregation in meiosis depends on the nature of translocation. When the individual is heterozygous for a particular translocation, one copy of the chromosome will be normal and the other copy will have the translocated segment. Similarly, these two chromosomes also have the same relationship. When such type of chromosomes associate, a cross-like structure is formed in case of a translocation heterozygote. In this structure, this copy of the chromosome has section which is homologous to these two chromosomes. Similarly, this copy of chromosome has two sections which are homologous to these two different chromosomes. Due to this relationship, a cross-like structure is formed in case of translocation heterozygote. So friends, this is all about different types of chromosome rearrangements. Next we shall discuss about aneuploidy. So continue to watch my YouTube channel, Sam Biology Class. Goodbye and good night.